The title of my project is The Effective Stages of Ripeness on the Amount of DNA from Kiwi Fruit. The testable question is, how do the different stages of ripeness of kiwis affect the amount of DNA harvested? My levels of IV are ripe kiwis, unripe kiwis, and overripe kiwis. The number of trials are 2 for each kiwi. My independent variable are the different stages of kiwis. My dependent variable is the amount of DNA harvested, and my constants are the amount of kiwis used. My hypothesis is if the ripeness of kiwi fruit affects the amount of extracted DNA, then the ripened kiwi will produce more DNA extracts because kiwis produce more enzymes, which help in breaking down cell walls, making it easier to extract the DNA. So if the kiwi that has more enzymes produces the most DNA. My materials are 2 ripe kiwis, 2 unripe kiwis, 2 overripe kiwis, pineapple juice, a tablespoon of salt, dish soap, cold isopropyl alcohol, 2 plastic cups, strainer, a bowl, a measuring cylinder, a paper towel, chopsticks, knife, a fork, and a chopping board. My procedure is I take 1 plastic cup and mix together about 80 milliliters of water, half a teaspoon of salt, and 2 teaspoons of dish soap. This makes your extraction solution. Next, you peel your kiwis and chop them into small pieces, then add the chopped up kiwis to a second cup and use a fork to mash them up. Then you add the extraction solution to the kiwis and leave at room temperature for about 20 minutes. Put a paper towel and a strainer over a bowl and drain the mixture. Transfer the liquid in the bowl to the measuring cylinder. Add the pineapple juice to the liquid, then leave at room temperature for about 5 minutes. After about 10 minutes, you should be able to see kiwi DNA. If you add the pineapple juice, at the top, you can take this out using a chopstick. Do this with the ripe, unripe, and overripe kiwis and compare the amounts of DNA you get. My data table is the mean DNA for the 2 unripe kiwis was 4 milliliters. For the overripe kiwis, it was 5 milliliters. For the ripe kiwis, it was 6 milliliters. This is my my table that shows the trials and data about how much DNA is extracted. My data analysis is graph number 1 shows the effect of different stages of kiwis that they have on their DNA. The mean DNA of the kiwis increased the more ripe they were. For example, the mean of the unripe DNA is 2.5. For ripe kiwis, the mean DNA is 4, and for overripe kiwis, the mean DNA was 6. This shows that the riper the kiwis were, the more DNA they produced overall. My conclusion is the purpose of this experiment was to investigate the effect of kiwis' DNA on ripe, unripe, and overripe kiwis. The results showed that the mean of the the results showed various effects. The amount of DNA increased when the kiwis were riper. The stages in kiwis caused a big difference in the amount of DNA extracted because of the amount of enzymes that they have. So that varies with the amount of DNA that they can extract.
Hello, my name is Julia Molina, and today I am going to explain my experiment. In In my experiment, I tested the pH of vinegar and water copper solutions' influence on the copper corrosion on pennies. I had a control group where one of the test tubes had pure distilled water and another had pure white vinegar, each with a copper test tablet. This is the tablet, and then these are the tubes that I used. This helps me compare my results from the 3 trials to the control group. So as you can see, in the copper is 0 ppm, and as we said, that was the water color. It was orange and reddish, so it represents 0 ppm. And then for the vinegar, it was 0.5 ppm because of the color, so it was more focusing on iron. In trial 1, the copper water solution ended up with 4 ppm of copper quantity, and the color was blue, as well as the copper vinegar solution. In trial 2, the copper water solution ended up with 0 ppm of copper quantity, which was orange and reddish. So this, after, it ended up with 0 ppm. But the copper vinegar, it ended up with 4 ppm of copper solution, which was the color blue. So as we can see, in trial 2, the only thing that changed was the copper of water. It remained at 0 ppm, while in trial 2 and in trial 1, the vinegar still remained the same as 4 ppm in copper. In trial 3, both the copper and water solution and copper and vinegar solution ended up with 4 ppm of copper quantity, which was the color blue. Additionally, after removing and rinsing the pennies from the designated solutions, the pennies that came out of the vinegar solution ended up cleaner than the ones from the water solution. So overall, the vinegar not only helps clean pennies better, but it also is more reliable to quantify the amount of copper corrosion found within the pennies. 2 of 3 times the water solution quantified a larger amount of copper corrosion, which is not as reliable compared to the 3 out of 3 times that it was quantified in vinegar. So we can see that overall, the vinegar not only helps to clean pennies, but it's also reliable. And that is the data that I got from my experiment. This is me, Julia Molina, signing out.
My name is Miazga.
My name is Jane Giles.
And today we're going to talk about average pH levels of everyday waters.
To keep with the project, we filled up each cup of water with each variety of water. For the next step, we're going to fill each water with the pH level testing drops.
We're going to fill each cup with 5 drops.
After waiting a few seconds, we should be able to mix it. Now that we're done with that, we're going to calculate each of the pH levels. The salt water, if we can see over here, then we can see that it's a 5 on the chart. So we're going to calculate that as a 5.
Put that in the calculator.
For the next one, we have the tap water. As we can see over here, it looks to be a 7. For the next one, we have the bottled water, and if we look over here, it looks to be a 6. Next one, we have the electrolyte affiliated water, and it's an 8. And for our last and final one,
We have the filtered water.
we have the filtered water, which seems to be a 6.
And if we add everything up, it'll be a 32 average.
And then after that, we have 5 bottles. We have 5 cups.
So we divided by 5.
We divided by 5.
And our total is 6.4.
And that is close enough to our hypothesis,
Which was 6.5.
This is class 6E7. Our project is all about bath bombs. The question for our experiment is, does adding more cornstarch affect the fizzing of the bath bomb? This is class 6E7, and our project is all about bath bombs. The question for our experiment is, does adding more cornstarch affect the fizzing of the bath bomb? This is our background research. We are researching to find out more information for our experiment. We found out that cornstarch makes the bath bomb fizz for a longer time. Our hypothesis is, if we add more cornstarch, then the bath bomb will fizz for a longer time because cornstarch will slow down the reaction. This is our investigation design diagram. Our independent variable is the amount of cornstarch. We had a normal recipe and a recipe with extra cornstarch. We had 3 trials for both types of bath bomb. Our dependent variable is the amount of fizziness. Our cornstarch, water, food coloring, thermometer, stopwatch, baking soda, expert salt, the types of cornstarch and oil. Our materials are food coloring, 3 containers, water, expert salt, baking soda, citric acid, bath bomb mold, cornstarch, stopwatch, thermometers, paper towel, kettle, and vagrants. First, we mix the dry ingredients for the normal and extra cornstarch recipes. Then we combine the wet ingredients for the normal and the extra cornstarch recipes. Now we combine the wet with the dry ingredients. We have to be very careful because we don't want the ingredients to react too soon. We place the mixture into molds and leave it overnight. Then we test the bath bomb by dropping them into warm water and seeing how long they will react into the water. This is our data table. On average, the normal bath bomb reacts for 1 minute and 56 seconds. The bath bomb with extra cornstarch reacts for 1 minute and 28 seconds.
Go.
Our graph shows us that the extra cornstarch fizzes for a lot longer than the normal bath bomb. Looking at the data, the normal bath bomb took a shorter amount of time to fizz. The average for the normal bath bomb is 106 seconds. The bath bomb with an extra cornstarch has extra fizzing. The average for the extra cornstarch is 268 seconds. In conclusion, our hypothesis was right. With the extra cornstarch, the bath bomb fizz for a longer time. In that normal recipe, the bath bomb did not fizz more. The cornstarch slowed down the reaction, which made the bath bomb fizz for a longer time.
The effect of different foods on the amount of glucose. How do different types of food affect the concentration of glucose? For my hypothesis, I predicted that fruits would have a higher concentration of glucose because glucose is the byproduct of photosynthesis. To support this in science bodies, it's stated, technically, sugar is a carbohydrate that occurs naturally in every fruit and vegetable. In addition, fruits might also have a higher concentration of glucose because it is the only food that has natural sugar. Many other foods, such as sugary drinks and juices, have added sugars. So glucose is a type of sugar. It is used to fuel brain cells and is an important source of energy in the body. Most of the glucose in your body comes from carbohydrates, which are long chains of sugar molecules. This was more background information about each type of food that I tested in the experiment. For the IDD chart, the independent variable was the different types of food being tested. The levels of IV were fruit juices, fresh fruit, processed foods, and miscellaneous, which consists of sugar, water, and honey. Each food was tested for a number of 3 trials, and the dependent variable was the amount of glucose concentration. The constants were the same test strips used to test the amount of glucose and the same 30 seconds to wait after dipping the test strip into each food. These were my materials. So the first part of the procedure was to test the test strips and to make sure that they were working properly. The second part of the procedure was to test each food using the test strips. These were my data tables for the fresh fruit juices, miscellaneous, and processed foods. These were my graphs. Based on the data tables and graphs, I noticed that fruits had the least amount of glucose concentration with averages between 0.25 and 2%. On the other hand, processed foods had a higher amount of glucose concentration measured. The purpose of the experiment was to find out which group of food had the highest amount of glucose. After conducting the experiment, I noticed that processed foods had the highest amount of glucose, therefore not supporting my hypothesis. During the experiment, several mistakes could have affected the outcome of the experiment. Pressing a test strip against the freshly cut slice of fruit was not the most efficient way to measure the amount of glucose in each fruit. The test strips might not have had a sufficient amount of juice to calculate the exact measurement. In addition, when preparing the sugar water, there was not an exact ratio of sugar to water that I had to add. This could have also affected the results because I might not have added enough sugar for the strip to measure it. To improve my experiment, extracting juice out of each food and creating a specific measurement of sugar and water could help get more accurate results. This experiment could be connected to real life because people with diabetes use these test strips to measure the amount of glucose in their body. This experiment could be led to further study of what foods have what amount of glucose. This was my literature cited. And these were my photos. These photos show the food that I tested, and these photos show the first part of the procedure in which I prepared glucose water to make sure that the test strips were working correctly. These photos show the glucose water that I prepared. I used food coloring to show the amount of glucose in each cup of water. These photos show the results of the first part of the procedure. Using the color on this test strip bottle, I made sure that the test strips were accurate. And these photos show the second step of the procedure in which I tested each food. In these photos, I pressed the test strip against a freshly cut slice of fruit. And here, I dipped the test strip into the rest of the foods. And these were my final results for the fresh fruit, fruit juices, and miscellaneous. As you can see here, the processed foods had the highest amount of glucose shown by the colors on these test strips.
The effect of hand soap brand on the inhibition of growth of microorganism. Question: how does the brand of hand soap affect the growth of inhibition of microorganism? Investigation design. Levels of independent variable. Type of hand soap. Dependent variable. Microbial growth. Hypothesis. If the brand of hand soap affect the growth of inhibition of microorganism, then using antibacterial soap will help inhibit the growth of microorganism because soaps play an important role in removing and killing bacteria. Materials for the experiment. Antibacterial hand soap, normal hand soap, baby hand soap, and 4 agar plates. Data table. Number of bacterial colonies. Graph. Here we can see how many colonies of bacteria are left after each soap. Analysis of data. The effect of the brand of hand soap on the growth of inhibition of microorganisms is summarized on Table 1. The agar plate that was treated with antibacterial soap produced an average of 43 colonies. The agar plate treated with normal body soap produced an average 14 colonies. The agar plate treated with baby hand soap produced on average of 40 colonies. And lastly, the control setup produced 55 colonies. Conclusion. The purpose of this experiment was to investigate the effect of various kinds of soaps on microbial growth. The antibacterial soap did not inhibit the microbial growth as compared to the normal body soaps, which showed a lower average amount of colonies with an average of 14 colonies. The mean average of colonies using baby hand soap was 40 colonies. The control mean average of colonies was 55 colonies.
So it's a 704 science investigation. You ready?
How will time update for other for level? And coordination of research based on tools will be more involved. In the daytime day, it will be less or at dusk.
The otters, right? Because that's what we were doing, the otters.
Ah, right.
So that's all the food that they eat, remember? Yeah, good job.
Ah, right.
The activity level of the North America river water increases in the
morning.
morning.
Good job. Okay, so what's this? What do we call this chart?
Investigation design diagram.
Good job. So how many times did we observe the river otters in the morning? What's the number there?
3.
3, good. And what about the afternoon?
3.
Good job. Okay, so what does this say?
Data for a visual return.
Okay. What was the number for where it says alert? What do our bars say?
14.7.
Okay, good. And that's for the daytime or the nighttime?
Daytime?
Good. What about locomotion?
18. Nighttime.
Okay, what about the not visible?
6 or 9.67.
Good. For the not visible, were they more visible at night or in the day?
Night.
So what's our claim say? Go ahead, you read one, Benson. What's the claim?
They're a more intuitive in the morning than they are at nighttime.
Good job. Hong, do you want to read the evidence?
Yes.
Go ahead.
This, according to the data analysis, the
frequency
of specific
behaviors
others engage in increases during the morning hours.
So I'm going to read the first part. Talk about some sources of error discovered and next steps you would incorporate if you were to run this investigation again.
Hello guys, I'm Ashley Campos from 75 and I will be conducting an experiment called the effect of different types of shampoos on hair. In my hypothesis, I state that the organic shampoos will be more effective and will be the best on the hair and skin. I believe this because on Science Buddies, they state that customers buy shampoos to clean their hair good and for it not to be greasy. The most common thing found in shampoos is to help clean the surfactants and for the hair to be cleaner. The surfactants is the part of the hair where there is grease. At the end of my project that I conducted, it was to test which one was the most compatible with the skin and hair. Different levels of these shampoos were on level where they are supposed to be processed with the hair and how they affect the hair. The store-bought shampoos were the most effective on the hair to remove the grease and dirt. My hypothesis was incorrect with me stating how the organic shampoos are the best to use on hair and skin. I believe that the organic shampoos were more efficient than the store-bought shampoos. In the end, the store-bought shampoos were compatible with both the hair and skin, instead of the organic shampoos. Therefore, my hypothesis was incorrect. This project was conducted by Ashley Campos from 75.
The effect of emotion on heart rate. The testable question is how someone's emotions can have an impact on their heart rate.
In this experiment, we decided to test the 4 mood meter colors, which are yellow, red, green, and blue, and see how they impact the heart rate. We decided to do this experiment because we know our school is mainly focused around social and emotional learning, and we think it would be a great way to see how it impacts us throughout our day.
The background research for this experiment is that different emotions have an impact on our actions. We know that if different emotions affect heart rate, then the color red in the mood meter, which is represented by anger, anxiety, highly stressed, mad, and upset, will produce a higher heart rate because according to the website, the effect of heart rate on your emotions, it stated that madness can increase the demand for blood. We know that when we're angry, we could have impulse decisions, making our actions much less controlled.
So we hypothesized that the more prominent emotions such as anger and happiness will have a bigger impact on your heart rate since the brain sends signals depending on how you feel, and those emotions give you the most reaction that sends the most blood and oxygen throughout your muscles. When you feel scared, anger, excited, your brain sends signals to your heart, automatically telling it to beat faster. So based on all our research and in the article that we found, Why Does My Heart Beat Faster?, we hypothesized that
that depending on the emotion, it will have an impact, and we believe that red, which is anger, will have the biggest impact. Our materials for this experiment is a technical device such as a computer or phone, a range of people, specifically 24 people, with a different range of ages, Zoom or any video call device, a heart monitor, and a notebook to write down all our information. First, we will prepare each person and put them into 6 separate rooms depending on their category of how they feel, which is red, yellow, blue, and green. Based on these, we will play a song up to each group and test the before and after effect on their heart rate. Then we will record all the data and compare them to how they reacted. So our IDD chart, our independent variable is the emotion, basically the mood meter. Our dependent variable is the heart rate, and our levels are the different emotions. It could be gloomy, content, joyful, or rage. We will try this 6 different times, and our constants will be the monitor, positions, and gender. In order to test our hypothesis, we decided to play upbeat music and see how every group's heart rate would react. As we can see on the table, each one is pretty designated to depending on their group, and yellow and green are the most average. After playing the music, we realized that the mood meter color red, which produces anger and frustration, had the highest heartbeat average. From this, it supports our hypothesis that when you're angry, your heartbeat will be faster. So based on the data we just analyzed, we noticed that almost every single group and every single person in each group had an increase in heart rate, except for a few. And even if it did not increase, it was very close to the before heart rate. So we believe that it did either have an effect or it didn't have much of an impact at all. To conclude our experiment, our hypothesis is correct that the mood meter color red, aka frustration, will have the biggest impact on the heart rate according to our data table and data chart. Since when we're enraged, our heart rate tends to go up along with your anger, yet when you're in the blue, your mood tends to give you a sad feeling and emotion, which slow down your heart rate. Basically, to end our experiment, we want to say that our hypothesis is correct, that once you are angry, your heart rate beats faster.
Our project was on the effective distance on the intensity of light. Our testable question was, how does the intensity of light change with distance? Our hypothesis was that if the distance that light travels affects its intensity, then where the light source is farthest away from the photoresistor, the resistance will be higher than rather feels closer. According to this website, the intensity of light diminishes with distance in accordance with the inverse square law. As light travels a greater distance, its photons are more widely spread out, and that causes its intensity to decrease. Our background research was, as you move away from the light source, the light gets dimmer. You most likely notice this with lamps, streetlights, and so on. You can measure this by using a light-sensitive resistor called a photoresistor, which has an electrical resistance that changes with exposure to light. You can use a digital multimeter to display this resistance. When the photoresistor is exposed to more light, its resistance decreases. This website states that in the dark and at low levels, the resistance of a photoresistor is high and little current can flow through it. A photoresistor's resistance increases as the light it's exposed to, it decreases. Our materials that we used were a photoresistor, a digital multimeter, 2 alligator clip leads, a lamp, a light bulb, a tape measure, tape, a cardboard box or another steady platform, a dark room, and a lab notebook. Our independent variable was the distance between the photoresistor and the light source, and our levels of the independent variable were 50 centimeters, 100 centimeters, 150 centimeters, and we did 3 trials for each of these. Our dependent variable was the intensity of light, which is displayed on the multimeter in ohms. Our constants were the same amount of external light present, ideally none though because it would affect the outcome of the project. We also used the same materials for each trial and the same light source. To summarize our procedure, we configured the multimeter to measure the photoresistor's resistance. We plugged the black and red multimeter probes into the corresponding ports as well as connected the multimeter probes to the leads of the photoresistor with alligator clips. Following this, we set the multimeter to measure resistance within the 200 ohm range, switched the multimeter on, and set the experiment up in a room without windows to avoid as much external light as possible. We leveled the photoresistor with the lamp and measured the distance from the lamp. Ultimately, we measured the resistance of the photoresistor as we increased the distance from the light bulb and took 3 readings from each distance and proceeded to calculate the average. This is our data table. Our data was all measured in kiloohms, which is about 1000 ohms. For 50 centimeters between the photoresistor and lamp, we got an average of about 52.33 kiloohms. For 100 centimeters, we got an average of about 167.6 kiloohms. And lastly, for 150 centimeters, we got an average of about 306.4 kiloohms. This is our data table, which shows the average resistance over distance. We observed that as the distance between the photoresistor and the light source increased, the resistance increased as well. Due to this, we can conclude that the intensity of light the photoresistor detected declined as distance increased. The multimeter displayed greater amounts of resistance as we moved the photoresistor and the light source farther apart. For instance, when the light source was 50 centimeters away, the average resistance was 52.33 kiloohms. When it was 150 centimeters away, the average resistance was 306.4 kiloohms. From this, you can infer that the intensity of light diminished with distance. We can conclude that our hypothesis was supported by the evidence we gathered. We observed that light intensity decreases as the light travels greater distances. However, there were some mishaps. We had to disassemble a lamp to use as the light source. We had difficulty keeping it in the same position, and if we did this project again, we would find something to hold it in place. Another challenge we faced was getting a completely dark room to allow the photoresistor to detect as little ambient light as possible. We found the knowledge of light intensity crucial due to how many different ways light is used. From eye surgery to disinfecting surfaces, light is extremely versatile and has an infinite number of real-world applications. This was the literature we cited during the project. And on the left is where you can see the digital multimeter and the small item connected to the alligator clip leads. This is a photoresistor.
My experiment is the effect of the amount of pressure on a gas's volume, which is a recreation of the Boyle's law. The Boyle's law was proved by an experiment performed by Robert Boyle, who was a chemist and physicist. My testable question is, how does the amount of pressure applied on a gas held at a constant temperature affect the gas's volume? My hypothesis is that if the amount of pressure applied on the gas held at a constant temperature affects a gas's volume, then I believe that applying more pressure to a gas will decrease its volume. According to this website, when pressure is applied to any object, the molecules' distance between each other will be reduced. When reducing that distance between the molecules, the volume will decrease. There's also an inverse relationship between the pressure and volume of a gas, so when molecules collide with the walls of the container they are held in or with each other, the pressure will increase. This means that the volume will decrease. Background information on the Boyle's law is that it was proved by the experiment performed by the famous physicist Robert Boyle, and it is a relation concerning the compression and expansion of a gas at a constant temperature. This law relates to outside topics like us breathing. We increase the volume of our chest cavity, creating low and high pressures in our lungs by inhaling and exhaling. These are the materials used to create the experimental setup. Our independent variable is the amount of pressure or the amount of bricks applied to the gas or setup. Our levels of IV are 1 brick, 2 bricks, and 3 bricks. Our dependent variable is the volume of the gas, and the constants are the experimental setup as well as the initial volume of the gas, weight of each brick, and temperature of the gas. This is the procedure which we follow to build the model. And in the model, we blocked the syringe opening with silicone to prevent any gas escaping. Our results showed that at 1 kilogram, the initial volume decreased by 9 milliliters around that value. At 2 kilograms, which is a bit more pressure than 1 kilogram or 1 brick, the initial volume decreased by around like 12 milliliters. And at 3 kilograms, the initial volume decreased by half of its amount, which was the greatest decrease in the gas volume. We could also see that in the graph. So based on the table and the graph, the higher the amount of pressure put on the gas, the lower the volume of the gas gets. So we observed that putting 1 kilogram on the gas, the least amount of pressure, the gas's volume was greater than the other two on each trial and in the average. So when we put the greatest amount of pressure on the gas, the volume of it decreased the most from the initial volume of 30 milliliters. Based on our collected data, my hypothesis was proven to be correct. The greatest amount of pressure put on the gas, 3 kilograms, caused the gas's volume to decrease by the greatest amount, going down by half the initial volume. This caused the trial to have the smallest gas volume as well, compared to the other two weights or amount of pressure. I believe this occurred because the smaller the amount of pressure put on the gas, the more spaced out the molecules are. If I were to recommend this experiment to other students, I would recommend changing the independent variable. They could test how the temperature a gas is held at with a constant pressure would affect the volume of it, which is also known as the Charles law, because the temperature could affect a gas's volume and how the molecules react to that. These are our websites used, and that's us performing the experiment. Thank you.
How does music affect our mood? Hypothesis. We predict music could change your mood based on the genre of music you are listening to. For example, if you're feeling upset or down, listening to hip hop or pop music can shift or change our mood to upbeat or yellow. We predict that if a song matches your mood, it can boost that mood. For example, if you're feeling happy or excited, songs from rock, pop, rap, or in yellow song can make you feel more energized. Independent, verbally different genres of music depend verbally the part, mood, or their change in mood. How will we test our hypothesis on how music will affect our mood? We will test our hypothesis by having students listen to different genres of music, pop, rock, and hip hop, and then checking for what mood it brings out of resembles. We will test our reaction to the song by doing a mood meter check-in before and after the student listens to each song.
Procedure. 1. During different times of the day, students subjects listen to a song. 2. The song genre varied hip-hop, pop, and rock. 3. Students express how they feel before listening to the song. Students provide language based on the mood meter. Okay. Analysis of data. Data collected 1.1 shows the overall mood change of the subjects before and after listening to a song. The mood meters reflect changes in energy as you travel up the y-axis. The mood energy rises as you travel down. The mood meter energy decreases. The dial in the results we generalized yellow/green was pleasant moods and red/blue were unpleasant moods. Subjects who stayed in the same color content yet felt an elevation in that mood after the song were generalized to a more pleasant/energized or more positive label. Okay. The graph 1.2 shows the pop music created an average percent of 72.5 positive percent change in mood as students reported a higher energized mood on the mood meter after listening to the songs from that genre. Conclusion. The purpose of this investigation was to investigate the effects of different types of music genre on our mood/emotion. We found that pop music created an average percent of 72.5% positive change in mood, rock music created an average percent of 55% change in mood, and hip-hop music created an average of 75% change in mood. According to these results, hip-hop music creates the strongest average percent change in mood. However, all the genres tested saw some average % of change towards a more positive mood. Factors that could have also affect the subject's mood were the time of day and the lyrics of the song. The data supported the hypothesis that different music genres affect our mood and that can boost our hypothesis. We would like to recommend to conduct further study on their music genre to determine the effects of mood/emotions.